Thank you so much for the organisers for inviting me to talk here. I only arrived for yesterday evening, but I'm around today, and I'm looking forward to getting more involved today. And I wasn't quite sure what I should talk about for my slot here. And so in the end, I just decided, well, I'll talk about the stuff that I enjoy most, which is doing the research that I do, and giving you a bit of a flavour for how I got to be where I'm at and where I've come from. So this is my lab here. We work at the University of Sussex. And in our lab, we're trying to understand how we remember our everyday experiences. So this is an example of a holiday I went to in Venice quite a long time ago, because my son here is a lot bigger than he is there. And when I think back to situations like this, I'm able to remember a series of events that happened, and they are kind of, I can bring to mind what we were doing while we were there. Sometimes, although my imagery is not very good, I can remember what things look like, and sights and sounds and things. And so our lab is trying to understand how these memory processes work in the brain that allow us to be able to remember these kind of rich experiences. And partly why I'm putting this in a film reel is that within the lab, rather than just giving people, say, lists of words to remember, we are using more often quite rich stimuli like videos to test memories for things which have a lot more going on in them. So a bit more like these day-to-day experiences, which have a richness that you don't get if you're just looking at sort of single words or pictures. So many of us, probably most people in the room, are able to create memories like this, and we're able to remember back to experiences that we've had. But of course, not everyone is able to do that. And you can think of this as an additional round of the pub quiz from yesterday. These are all individuals, either fictional or from real life, who have severe memory problems. And I won't go through them all, and I won't test you on them, but we've got Dory here. Finding Nemo, and Dory, the fish from Finding Nemo, is one of the best on-screen examples of what it's like to have very severe memory problems. And it's quite a sensitive and accurate portrayal of what it's like. Memento is kind of less accurate, but it's a really great film. Henry Molaison was an individual who had a very, very severe amnesic syndrome and took part in studies of memory for the whole of his life, and we've learnt an awful lot about how memory works and how it can go wrong through the studies that Henry took part in, as well as people like him. And I am going through all of them, I thought I wouldn't. And we've got Iris Murdoch here and Ronald Reagan, both of whom had Alzheimer's disease and associated memory problems. And the interesting thing to me about these individuals with memory problems is how selective it can be, and what a separable process memory can be from other things. And I mean, Ronald Reagan, there's anecdotes of him being able to reproduce some of his famous speeches. He could get all the intonation, he could talk a lot more fluently than I can, and deliver a fantastic speech, and then sit down and minutes later have forgotten the fact that he just stood up and gave this speech to people. So you've got other things like attention, the ability to hold information in mind in the moment, your access to language. It can all be very, very well preserved, and yet you still have a devastating impairment in your memory. And for me, that is one of the most interesting things that I get to look at, is not only how memory works in the brain, but how it can go wrong in the context of brain disease. Okay, so how did I get here? I'm going to do a one-slide whiz through my life up to the point where I'm standing here. I was doing science and maths as A-levels. I wasn't doing biology, and I wasn't particularly interested. I was finding that I wasn't particularly engaged by the stuff I was doing. I wanted to go to university, but I didn't know what to study, and I went back to my old biology teacher and said I was thinking about psychology, and he had suggested that I read this book that I'd never heard of, which probably most people have heard, The Man Who Mistook His Wife for a Hat by Oliver Sacks. And I read it, and it's about what we now call really neuropsychological case studies, people who've had brain damage, and that's left them with some specific impairments in how they think in different ways. And I thought this was amazing, and based largely on that, I decided I definitely wanted to study psychology. I went to Oxford and did experimental psychology. After that, I took an assistant psychologist position at the National Hospital in Queen Square. And there I got to work with some of these people who had similar syndromes to those that Oliver Sacks had described. And indeed worked with a very densely amnesic individual VC, and this was a paper that we published where I carried out research. I was the research assistant who went on and did a whole load of memory tests with this individual. Following working at the National Hospital, I then switched sides in Queen Square and went to work at the Institute of Cognitive Neuroscience, where I did my PhD. And then I went to Sussex to become a lecturer. And that sounds like a very logical stepwise progression, and it didn't feel like that at all at the time, and that was never it's interesting to look back and sort of join dots in your past. But I've always been interested in memory and how memory can go wrong. And so that's kind of how I've got to where I am now, working at Sussex. And what I wanted to do in this talk is to just spotlight 3 research studies that I've been involved in, and how they've really continued this interest in particularly how memory can go wrong and understanding memory in the context of brain disease. And so the idea of this is just to give you a flavour for the kind of research that we've done, what things that we can conclude, but also some broader points about what this has taught me about being a researcher. So we're going to start with spatial memory. So I don't think I can click on this, but if you could click on the video to set this off. This is, the video is showing a speeded up version of actual data collected from a rat that was foraging around a circular arena while it's got an electrode implanted into a part of the brain called the hippocampus. And the noises you're hearing is when a cell in the hippocampus, one specific cell that is being recorded from, is active, and the red dots show the location of the rat when that cell was active. And what you can see is that these dots all cluster in one particular part of the environment. And so these cells are known as place cells because they seem to signal to the individual, the rodent, where it is within this environment. And this work was originally pioneered by John O'Keefe at UCL, for which he and then Maybrit and Edvard Moser got the Nobel Prize for understanding how space is represented in the brain. And so, these basic science experiments have revealed a lot about how the brain seems to signal where you are within an environment. In fact, we're in a circular environment here. This would feel quite similar to that rat. And if we move around, then I'm pretty sure there would be cells within our hippocampi that would fire when we're in specific locations within this environment, because our hippocampus does a similar thing. If you look at things like spatial memory, you're looking at behaviors that are preserved across different organisms. Another tip for anyone doing a presentation is have water with you because your mouth gets very dry. Okay, so we wanted to build on from this. This is when I was working with Neil Burgess at UCL. and wanted to see whether we could create a new test that would be based on our knowledge of how the hippocampus in particular seems to represent spatial information. And that's important because the hippocampus here, particularly here, is involved in very early stages of Alzheimer's disease. It's one of the earliest places where you get amyloid deposition and you get neuronal death. So this, if you can develop a test that is specifically targeting functions of this region, then it might be particularly sensitive to the earliest changes that occur in Alzheimer's disease. And so the test that Tom Hartley devised with Neil Burgess is called the Four Mountains test. And this shows a kind of heat map layout of the topography of a computer-generated scene. And when you render this, you can make pictures that look at this sort of very empty-looking landscape. And this means that we can really get at whether people can process the features of the landscape. And I'm going to show you what this is like in a test that is horrendously difficult in a sec. So we had a version where you just have to do this when you have the picture in front of you, so whether you can actually perceive the spatial information. And then we have the difficult one where you're shown a picture and then it goes away for a bit, and then you have to recognize the same landscape. But it's going to be from a shifted viewpoint. So you have to try and recognize what this place will look like if you were looking from a different direction. So here we go. Here's the landscape. Here you can see there's 1, 2, 3, 4 different mountains within this kind of plane. And the test is to look at pictures like this. And I'm going to give you a clue that you're going to see it from somewhere over this direction. And you have to match one of 4 pictures which shows the same layout of mountains as is shown in this one here. This requires quite a lot of concentration. Okay. So one of these shows that same layout. Who thinks it's this one up here? I'm going to see whether anyone is willing to put a hand up. This one here, this one down here, and this one over here. Okay, the majority got it correct. It is this one over here. So you do 15 of these items in the test. You're doing them with it right in front of you in a quiet room. It's quite different from this situation here. And this is just showing the results of a study that I carried out where we were looking at people with different types of degenerative conditions. And the important thing here is these group of people. So you've got people who had a diagnosis of mild cognitive impairment that was characterized particularly by memory problems, who were at high risk of converting to Alzheimer's disease, people with a diagnosis of Alzheimer's disease, people with some different types of neurodegenerative conditions, and a group of age-matched healthy controls. And these people were doing significantly poorer on this task. So it was looking like this was indeed sensitive to some of these early changes that you got within Alzheimer's disease. The test has been used by collaborators, and also different versions of spatial memory tests are being used more and more to look at these very, very early changes that characterize Alzheimer's disease. This is an interesting one. This is people who had a diagnosis of MCI, who then later either converted to a diagnosis of Alzheimer's disease, shown here, or did not. And again, there was this clear separation, I mean, a very clear separation in this particular study. And interestingly, the score on the test correlated with measures of tau in the cerebral spinal fluid. So it also seemed to be a fairly good index of a disease process going on. So my take home, my point of presenting this really was to talk about a couple of things. That spatial memory itself is a useful model system to understand how memory processes work, and also it has shown some potential for looking at these very, very early changes that characterize Alzheimer's disease in particular. And the other point as well is that, that people carrying out more sort of, they're all rather derogatory terms of basic science or fundamental science or pure science is less so, but less applied science. The insights from this can be very, very useful, taken forward and then applied in clinical settings. And I mean, I'm very much at the point of doing this more basic science research. But obviously, the insights from this can be useful for developing tools that be useful for diagnosis, for monitoring disease progression. If we're getting pharmacological treatments, then you want something that will accurately measure the outcome of these treatments. And so, there's a use for these kind of tools. Okay. So now I'm going to talk about research which is more relates to what my lab's doing now. This is a study carried out by Chrissy Odekoven, who was a postdoc within my lab. Has now gone back to Germany and is working in memory assessment clinics in Germany. And if we could set off this video, I'm going to just show you this. So we use videos as a way of, as I said, kind of as a proxy for real life experience. So you can watch things that have a structure to them, they unfold over time, and they make sense, and they're quite engaging, and we test whether people can remember them later. So this is one that we've used. And they're okay, it's okay at the end. So we've used these in lots of different settings, but in the study I'm going to talk about is we're looking at people who are at risk of getting Alzheimer's disease. So we're working with Sussex Partnership NHS Trust, and particularly with their memory assessment service clinics. And in this particular study, we were seeing people who were all first referrals to the clinic and had been referred on to our imaging centre for an MRI scan. And at that point, we were approaching people and seeing whether they would like to take part in our research. And that meant that we could then later divide people up according to their diagnosis. We didn't know what their diagnosis would be at the time. We just knew that these were people who were relatively healthy. They were able to take part in the research that we were doing. That's sort of cutting right onto the results. This is looking at how well people can recall details from these types of videos. And we've got some healthy controls that didn't come through the clinic. Then we've got people who have subjective cognitive decline. So they feel that their memory has got worse, but actually objectively, there doesn't seem to be any concerns. They are doing as well as the controls. And then we've got two groups of people with the diagnosis that they received of MCI, and then some of whom, within a 2-year window, then converted to a diagnosis of Alzheimer's disease. Other ones, as far as we know, have stayed the same or were discharged. And so we can see that this is very good at indexing their memory, and it's relating to this disease progression. You can also see, because we were scanning people while they were watching the videos, so this looks like a functional scan. It's actually not. This is looking at the brain structure and the amount of grey matter across the brain and how that correlates with their scores on this test. And what we can see is that there are many regions of the brain where the less grey matter that you have, the poorer that you do at remembering these videos. And they're occurring in parts of the brain, this wider network, which we know both to be involved in memory, but also is where we see changes in Alzheimer's disease. So this is very consistent with the idea that some of these, we've got a gradient of people at different stages of a disease process. Now, what we were also doing was scanning people with a functional scan while they were watching the videos themselves. And then we did a type of analysis that you often have to do if you're watching movies or videos, where you have to be a bit clever at how you analyze the data. And what we're doing is looking at how correlated brain activity is across different individuals. So the idea is if you're watching a video and there's a big loud noise, then in everyone's brains, in their auditory cortex, you're going to see an increase in activity because it's processing a big loud noise in everyone's brain. And so that will be correlated. You'll see the same kind of thing happening across all the people who are watching the same video. But it turns out that goes on right throughout the brain into areas that are much more involved in abstract things like making sense of a narrative as it's unfolding. And so we still, in healthy people, will see correlated activity while they're watching a meaningful movie. You don't if you're seeing just sort of jumbled up scenes from a movie. And here I'm showing areas that are less correlated in the people with worse memory. So while you're actually watching the video, these people in particular parts of the brain, some of whom we know are involved in Alzheimer's disease, but are also involved in making sense of situations, of understanding things as a situation unfolds. These are showing decorrelated or less correlated activity. So it implies that there's something going on that means that these people are not, even in the now, even while they're watching something, are still not kind of knitting together that information to create a coherent understanding of what's going on. And so we can infer from this that when people have poor memory, then part of that is because they're actually not encoding that information, they're not knitting it together in a meaningful way at the time. Mouse dry. And so more broadly, by carrying out studies like this, by having a better understanding about the specific processes involved in memory that are going awry, means that you can target compensatory strategies, ways of coping with memory problems more directly if you have a better understanding. Because of course, if you forget something, there's a whole load of reasons why you might forget it. But if we can pinpoint what processes are going wrong, then you can better support people. And also, I wanted to make the point, so when I was working at UCL, certainly as a PhD student, I was told by someone who was interested in clinical research but said, you're never going to be able to do it unless you actually work in a hospital, you're never going to get access to any patients, it's give up on that as an idea. And this just isn't the case, I found out. We have a really great relationship with Sussex Partnership Trust. It helps that we have lots of funding, but they support us in the research that we need to do and that we want to do. And so, I found that this can be very productive. Okay, so the last thing that I wanted to talk about is some more recent studies that we've been doing looking at the effect of ApoE on memory. So carrying an ApoE4 allele is the largest genetic risk factor for developing Alzheimer's disease. And around sort of 20 to 25% of the population will have at least one E4 allele, and the risk is if you carry one, there's an elevated risk of sort of 2 to 3 times. If you have two E4 alleles, then there's more like a 6 to 8 times risk of developing Alzheimer's disease. And we were interested in seeing whether there are any effects on cognition, on thinking and memory in particular, in healthy people who carry these alleles. So this was research carried out by Jess Daly, who is somewhere in the audience, who I can't see, who is a PhD student, and Claire Lancaster, who is an Alzheimer's Society funded fellow. And really they drove this whole project, and it's a lot larger than the snapshot that I'm going to talk about here. And Jess was funded by a doctoral training centre at the University of Sussex, which is funded by the Alzheimer's Society. And the broad aim is to look at the different ways that ApoE might affect cognition and why there's this link to getting Alzheimer's disease and also actually other brain conditions. And so people are looking at this at different levels, which is why it was, do I have the title? Of molecules, mice and men. So it's really at Sussex, we're not huge, but we have a broad area of research. And so you've got researchers looking at the same thing, but across very different levels of explanation. And so yeah, Jess was funded by this. Now, we had some ideas for how we were going to look at this, which would have involved bringing people to the university, taking cheek swabs, genotyping people, working very, very, very hard to collect what ultimately would have probably been a relatively small sample to look at effects that probably are not particularly strong in the healthy adult population. But then the COVID pandemic hit, and we couldn't do any of that anyway, and so we had to think what on earth we were going to do and how we would address some of our research questions in the context of a pandemic when we couldn't see anyone. And so fortunately, through Claire Lancaster's links with the NIHR Bioresource Organization, we were able to send out emails to have a link to an online web platform where we could give different tests that we were interested in a much, much larger population of people than we would have seen ever at Sussex, in genotyped individuals. And so we were given details of around 400 people through this, all who had their known genotype, and Bioresource actually then balanced our group. So we were trying to see as many people as we could of people who had 2 E3 alleles, so that's the sort of population norm, 1 E4 or 2 E4s. And thanks to particularly Sam Behrens in my lab, who was very good at setting up our online testing system, it meant that we could deliver tests of a complexity that I never thought we would be able to do online. I thought these were things that you'd have to bring people in and give people detailed instructions, and it turned out actually we could have this sophisticated web platform hosting the kind of tests that we wanted to administer, and people could do this from their own homes by just having a link sent in an email. So I'm first going to talk about a study that Claire led on, where they were using a test called the mnemonic similarity task. And here you're showing a series of objects, one after the other, and some of the objects are repeated, and you have to pick out the ones that you've been shown before. But the catch is that sometimes you'll be shown something that is very similar to what you were seeing before. So this is all showing the similar items. And you've got to distinguish similar items from the true repetitions, the one where you see exactly the same thing before. And this really involves a fine-grained distinction between these different objects, and is thought to rely on processes underpinned particularly by the hippocampus. So this part of the brain that we know that is involved early on in Alzheimer's disease. And turns out over here that the genotype had no effect at all on people's ability to do this task. So here we've got the double E4s, who are the most genetically at risk of having Alzheimer's disease, but there is no difference. And I'm just showing one of the outcome measures. This is your ability to distinguish between these called lures, the ones that look very similar to the ones that you've seen before. And what you can also see, all of these dots are individuals. We've got nice big samples, so we're able to really show that better scores is better memory. There's no difference across this at all, and in fact, there's good evidence that these are equivalent. So Bayes evidence that these are equivalent to each other. And then a task that Jess led on was a different type of spatial memory task. So here you're shown different objects against a background, and then first of all you're shown different objects and you have to say whether or not you think you've seen them before, and then you have to try and put them back to the location they were. So you use your keys to shift this around on a ring, and so you don't see the ring here, you just see your object moving around against the background until you think, yeah, I think it was up over there. So you're aiming to put your torch back into the same location that it was before. So again, this is rather different than the Four Mountains test, but it still requires you to have a knowledge of where something was within the environment, so what it was and where it was. And similar to the other one, this is showing two different measures, your ability to recognize whether you've seen the objects broadly in that location, and then your actual precision. So this is perhaps a more important measure of how accurately you can get it right back to where it was that you saw it before. And this is showing the different genotype groups. So you've got the EE3s, E3, E4s, and then the E4, E4s, so the people who are at highest genetic risk, but there is no difference across these at all. So this is, I think, quite good news for people who carry this particular gene, because it's saying that, really in healthy people, it doesn't seem to have any measurable effect on these tasks, but also we've looked at some other tasks as well. But I mean, these ones I'm picking out because we know that they should be quite sensitive to the changes in the part of the brain that are involved early in Alzheimer's disease, and they're not. So there is obviously a genetic link. It's conferring some kind of vulnerability to developing Alzheimer's disease later in life, but it's not changing things across the lifespan, as far as we can tell from this particular study. Okay, so obviously the broader aim of this is to try and understand what is linking things like genetics to the disease progression. And this is why it's important to have, let's say, at the DTC in Sussex, people who are looking at this in preclinical work and at the level of cells and the molecules, as well as the stuff we're doing, which is looking at behavior in healthy people. And also, I mean, if you were paying attention, you might have seen in the early study with the Four Mountains test, there are very few individuals involved. I mean, these are small-scale studies that you can do as part of a PhD, but in the context of sort of university research, we don't have huge resources, but also at the time we didn't have access to these fantastic things like the bioresource, where you've got large cohorts of people, and so it's being able to link up with different groups that has really pushed forward the quality of the research that we can do, and that is so important and is becoming more and more important, and the more linked together we are and the more we're sharing our resources, the better the research that we can do. We also pre-registered the analyses that we were going to do, so we're trying to say up front, what we're going to do with our data, how we're treating it, and I think also that is contributing to better quality science. And that is really all that I wanted to say, and I want to leave some time for questions if there are any. So just thanking the people that were involved in these particular studies, and also the organisations that I'm part of. But obviously there's this sort of wider group who are involved in all of the research that we do. And thanks to the Alzheimer's Society for their funding, and really thanks to the ERC who have funded my lab for what will be about 10 years altogether, and enable me to do research that is both looking in healthy populations, but as well dipping into the clinical research that I'm so interested in as well. Okay, that's the lot.